Hi, so thank you for the introduction. As you said, I'm Patrick. I'm from Feng Zhang's lab at the Broad Institute, and I'm really excited to be here today to tell you about our work on Cas9-based genome engineering technologies. So I'm going to keep this more of a general talk. This will be a sort of broader overview of what we're doing and what we're thinking about in doing CRISPR-based technology development. So a lot of this work was motivated by the need to precisely engineer biological systems. And being able to modify the genomes of any organism we want would be a really powerful technique with broad implications from basic biology to a new generation of medical therapeutics to biotechnology. This would, for example, allow us to begin to interrogate the genetic and epigenetic diversity underlying the nervous system. Or sometimes we would want to make a change and really see what happens. So you could imagine a future where you walk into a clinic and instead of taking a prescription drug that treats the symptoms of your disease, you could have a gene surgery, if you will, that would take the harmful DNA mutation that leads to your disorder and replace it with a healthy sequence. Finally, this has broad implications for biotechnology, from engineering biofuels to food security or generation of novel synthetic materials. The broad concept underlying the new generation of gene editing tools is the idea that we can hijack the endogenous repair machinery of the cell. And that is underpinned by generating a double-stranded break on the DNA within the genome. And what happens then is the cell will undergo a repair process because it'll say, SOS, my DNA has broken. I can either typically repair the DNA through a non-homologous end joining process or an homology-directed repair, or HR, homologous recombination-based repair pathway. Now, NHEJ is the more typical decision tree, and it's because it recruits these Ku-based proteins that have high affinity to double-strand break ends and attempts to stitch them back together. The problem with NHEJ, however, is that it's an error-prone process. So it leads to the introduction of short bases that are inserted or deleted at the double-strand break site, which can lead to a mutation. So if you imagine if this mutation happens in the coding region of a gene, this could lead to a frame shift mutation that would either scramble the protein or lead to a premature stop codon. So this is essentially an error-prone repair process that leads to loss of function or gene knockout. The other pathway, homology-directed repair, is a more high-fidelity pathway that allows you to introduce, remove, or make precise modifications within the genome. So it allows you to do precise gene editing. And the main difficulty with HDR historically has been the extremely low absolute frequency of HR repair. And so it turns out then, how can we stimulate repair by generating DNA double-stranded breaks? And the underlying sort of idea was that we could use targeted nucleases to facilitate genome editing, and the vision was, can we turn any organism into a genetically tractable model the way that we historically have been able to easily genetically engineer E. coli or yeast? And one of the main concepts was the separation of DNA binding domains and nuclease effector domains that can actually be recruited to the genomic locus of interest to generate a DNA double-strand break. And these DNA binding domains historically can come from a variety of methods, one of which is, as we're interested in today, derived from a mysterious prokaryotic system called CRISPR. And so these CRISPR systems are adaptive immune systems found throughout bacteria and archaea, and they originally experimentally determined to be active immune systems found in bacteria in yogurt cultures. And this has been converted into a really powerful tool for engineering biology in pretty much any genome that people have really tried to get it to work in. So what is CRISPR? How does it work? It turns out it's really an immensely complicated system that begins with foreign genetic challenge. So imagine a bacterial cell, and you have it's invaded by a bacteriophage or by a foreign plasmid via horizontal transfer. And what the CRISPR system does is it'll take these fingerprints shown in red and insert them into a CRISPR locus, which is present on the endogenous bacterial genome. And these fingerprints then become templates for later recognition and search and destroy processes. And so this is a generalized CRISPR locus, and basically we have a cluster of Cas, or CRISPR-associated genes, as well as the CRISPR array, which is what gives CRISPR its name, which is clustered, regularly interspaced palindromic repeats. And the repeats that the CRISPR name refers to are shown here as direct repeats, which are interspaced by spacer sequences. Then these spacer sequences are the fingerprints that are derived from the foreign genetic material. Now the next process is one called cRNA biogenesis. cRNA simply stands for CRISPR RNA, and it's a way of turning these fingerprints, which are derived from DNA and inserted into the bacterial genomic DNA locus, into short RNA guides. And this goes through a variety of different types of CRISPR systems, and we'll focus on the main type, type 2, which has the Cas9 nuclease that we're all most interested in. Briefly, this locus here, this CRISPR array, is transcribed as an RNA, which then is associated with one other non-coding RNA called a tracer RNA, which stands for transactivating CRISPR RNA. So these names are not terribly imaginative. But essentially you need these cRNAs, these CRISPR RNAs, which are these spacers, which associate with these tracer RNAs, which become a mature guide RNA that can direct the single Cas9 nuclease to the genomic DNA shown here in blue. And it does this via this fingerprint shown here in red, which can directly Watson-Crick base pair with the genomic DNA of interest. And the key here is if you look in the type 1 and the type 3 CRISPR systems, these are multi-protein complexes, whereas the Cas9 is a single effector nuclease with a dual RNA guide. So let's take a little closer look at the basic history of what happened. Back in '87, Ashino et al. Was a Japanese group that discovered these really strange repeats just by sequencing the IAP gene in E. coli. And the reason why this was an interesting report back in the 80s was because typically repeats in nature are found as tandem repeats going repeat, 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 while these repeats were actually spaced out and spaced by variable sequences. In 2000, Mojica, working in Spain, discovered that CRISPR was really present throughout all prokaryote genomes, and then over the next 10 years or so, the really basic building blocks were really well elucidated. First, Rodolphe Bengu, working at Danisco, which is the dairy production company, they were trying to figure out ways of generating more resistant strains of yogurt, elucidated the antibacteriophage function of type 2 CRISPR systems, which contain the Cas9 nuclease that we're interested in. And the way he did this was by challenging these different strains of yogurt bacteria with different bacteriophages, and found that they were able to uptake fingerprints and then aid in phage defense. Then in 2010 and 2011, there was a series of really seminal studies first showing that type 2 CRISPR cuts DNA, so it acts actually on DNA targets, that it needs three components, the CRISPR RNA and the tracer RNA, to fully maturate these RNAs into the guide RNA. And then finally here, Stapranauskas, working out of Lithuania actually, demonstrated that Cas9 is a sole nuclease responsible for cleavage activity by modularly porting CRISPR locus from Thermophilus into E. coli. They're able to show that these CRISPR systems were modular, which really inspired us to be able to try to move these into mammalian cells where they could potentially function independently. So if we take a look at the CRISPR locus from two different bacteria, one is Streptococcus thermophilus, the other Streptococcus pyogenes, I've already shown you the general organization of these loci. One is this cluster of Cas genes, the other is these direct repeats and spacers, and you have this Cas9 nuclease, which is involved in the processing and targeting of these spacer sequences. And so if we try to reverse engineer this complicated prokaryotic system in mammalian cells, we essentially can break down the building blocks into individual expression vectors. And so if you take the Cas9 nuclease and put it on one plasmid, you have another CRISPR RNA as well as a tracer RNA on a separate plasmid. And these, because they're short RNAs, they're driven by U6 promoters, which are RNA polymerase III promoters that are typically used to drive siRNA or shRNA transcripts, as opposed to the classic RNA pol II transcripts, which are used to drive typically things that encode for that become mRNA. Additionally, we had this one piece called RNase 3, which in prokaryotes was RNase that was required for proper processing of these RNA guides. Here, essentially see, this is the target DNA. This is a human EMX1 locus with a DNA protospacer, and the CrRNA, which is the RNA guide, so basically you can replace the fingerprints that come from the phage genome with the genome from the human DNA. So you could target anywhere within the human DNA of interest. And one thing that I really want to point out here is this purple motif called PAM, which stands for protospacer associated motif. And it's essentially the main restriction on the targeting space of Cas9. Now, one thing I want to say is that the Cas9 nuclease is found throughout the microbial diversity, okay? So different CRISPRs or different Cas9s from different species of bacteria archaea would have different PAM requirements. Because this PAM motif is required to be immediately downstream of the target DNA, you can basically switch out this 20 base guide RNA for DNA targets that have this motif already on the genomic DNA. For the pyogenes Cas9 that most people use in the literature, that's a very relaxed NGG motif, and we'll talk more about how that means later. So the first proof of concept, Cas9 can cleave endogenous mammalian genes, we were essentially trying to figure out what was the minimal component system that would allow us to reconstitute CRISPR in mammalian cells. And it turns out all you really need is the Cas9 nuclease, the tracer RNA, and the CRISPR RNA, and that would allow us to introduce indels. So this is an assay where we can read out after a population of cells have been transfected with these components and they cut the DNA, what is the level of the mutagenic indel repair? Okay, so let's summarize. Bringing CRISPR into mammalian cells. You have a Cas9 nuclease shown here as the Pac-Man, and you have your guide RNA. And so later it turned out that you could fuse these guide RNAs together. So the CRISPR RNA and the tracer RNA could be fused to make a single guide RNA, or sgRNA, which would reduce Cas9 or CRISPR into a two-component system, one with your nuclease and one with your guide RNA. And this could make double-stranded breaks at DNA targets, which would then be repaired by NHEJ, which is an error-prone method for loss of function, or HR, homologous recombination, which is a method for introducing precise modifications within the cell. Okay. So we're able to bring Cas9 into the human genome. We can replace these fingerprints that are supposed to be phage sequences with sequences from human DNA. So we're able to then make modifications within the human genome. But then what's really next? And how can we bring this technology simply beyond proof of concept? And here are some of the really core considerations that we're really fascinated by in the technology development area. One is, of course, improving the efficiency. So if you target Cas9 into a population of cells, what percentage of these cells are we able to modify? Can we make homozygous mutations, or would they be heterozygous? How does that efficiency change, and how can we maximize it? Understanding and improving specificity. So in my introductory slides, I alluded to the possibility to usher in a new class of medical therapeutics. However, of course, the specificity of these nucleases would be extremely important, because you could imagine if you introduce off-target modifications that could really be functionally deleterious. Inducibility, so could we gain not just spatial, but also temporal control, so that we could really start to dissect temporally dynamic processes such as gene transcription, trying out novel effectors. How can we bring this as a platform technology beyond simply genomic editing, the modification of DNA? Could we modify the transcriptional state of a cell? Can we modify the epigenetic state of the cell? This really here is really limited by your imagination and your engineering abilities. Orthogonal control and multiplexing. Can we make different kinds or multiple modifications within the same cell to look at not just single genes at a time, but multiple additive effects? Structure-guided engineering, we recently reported a crystal structure of Cas9. How can looking really deeply at this protein and how it interacts with the target inform our engineering abilities? finally exploring delivery mechanisms. Imagining if you want to do in vivo modifications, how can we actually deliver this protein and these RNAs into tissue? So we'll talk first about how single guide RNAs were first engineered. And so this is the CRISPR RNA and this is the tracer RNA. So this is the natural 2-RNA system that guides the Cas9 nuclease. And so Martin Genec, working out of Jennifer Dowda's lab at UC Berkeley, showed in 2012 that you can indeed fuse these 2 RNAs together to make a minimal single guide RNA. And what the first test that we did was to compare this natural system with the chimeric guide, and we found 19.6%, here is 7%, 0%. In fact, the natural 2-guide RNA system seems to be a lot more efficient. And this didn't really make sense to us, and we realized if you carefully analyze these sequences, you find that this design, which works to direct Cas9 to cut DNA in vitro, of course, didn't seem to work well in human cells. So what is the contribution of these secondary structures if you extend the tracer RNA sequence? And it turns out by generating a series of truncation mutations, we're able to figure out, here this is plus 48, plus 54, plus 67, plus 85. We generate truncations at different points along these RNA secondary structures, and we show by northern blot that in fact these hairpin loops seem to really, this is plus 67, plus 85, seem to really lend to contributing to stability of these guide RNAs. What's more was here plus 67, plus 85, you can immediately see that not only were they more stable, they also really helped you modify DNA. So up to 52% in general of the cells that were targeted were able to be modified. So that is really pretty good, right? 52% is much better than the single-digit numbers that we were getting previously. And what's more is by increasing the length of expression time of these reagents in your cell, you can really start to drive up these numbers to an as of yet undetermined ceiling. Okay, so now how can we understand or improve specificity? So I previously said that the PAM motif, which for S. pyogenes Cas9 is NGG, gives us a lot of target sites throughout the human genome. It allows us to target on average about 8 bases, every 8 bases within the human genome. However, the same characteristics that give us a lot of Cas9 possible target sites also gives you many possible off-target sites. Okay, and one way of getting around that is by converting Cas9 into a nicking enzyme. So it turns out Cas9 has 2 catalytic domains, each of which nicks a DNA strand. And the key was that by inactivating one of those domains, you can convert Cas9 from a nuclease, which generates DNA double-strand breaks, into a nickase, which generates DNA single-stranded breaks. And the key realization here was that single-stranded breaks of DNA are typically repaired with really high fidelity. However, Cas9 as a nickase could mediate HR. So by using single-stranded oligos instead of traditional long double-stranded homology donors that people use for generating knockout mice, you could introduce short single-strand oligos to get a certain amount of HR repair within the cells. So this is old data, and you can really start to drive these numbers higher. But initially, these numbers without nuclease stimulation or nickase stimulation would be around 1 cell in about 1 million. So this is already much better. Another thing that we did was we collaborated with the Yanish lab to see if we could inject these reagents into mouse zygotes to generate animal models. And so by taking zygotes, fertilized, so these are fertilized at the 1 cell stage, and microinjecting Cas9 mRNA, as well as guide RNAs targeting loci of interest, we were able to generate modified blastocysts at multiple loci. And then following blastocyst transfer, you could quickly generate double mutant mice. And this really was a rapid production of transgenic animals that really takes, really 3 weeks to get your first set of modified transgenic. Secondly, by introducing these homology donors, you could really make mutant mice with predefined precise mutations. And people have already been able to modify over 8 alleles at a time. So that's been really exciting. One outstanding problem with generation of these transgenic animals is the introduction of genetic mosaicism, because you can cleave the DNA within these single-cell zygotes. But if you imagine if the repair happens at the 2, the 4, even the 8-cell stage, then you could get multiple genotypes when these blastocysts expand to make a full embryo or into a full animal. And so this still requires then to get a pure genetic background, an isogenic background rather, you would need to have some kind of back crossing. But I think genetic mosaicism is a technical problem that will be solved in the coming months. So back to Cas9 specificity. So if you think about it, you have this guide RNA, okay, that directly wants to create base pairs to your target DNA. And so this is presumably some kind of interaction that may be governed partly by the Cas9 protein, also partly by nucleic acid thermodynamics of base pairing. And so the experiment that we wanted to do was to figure out how many mutations can you have in your target DNA or in your guide RNA. And the assay that I did was to say, okay, I have a 20 base guide RNA. Let's generate a library of mutants that have all possible base substitutions at every position along the guide RNA. This would allow us to investigate not just the positional effects of mutations, but also what's the effect of particular types of base switches, right? And how could that help us understand any thermodynamic contribution to off-target effects? And so if you look at these heat maps, I'll just briefly orient you. This is the original guide sequence, 20 bases, and these are the sequences that you mutate the guide to. And here the heat map is 0 is white, 1 is blue. It's normalized to cleavage, so 1 is cleavage. And so essentially you see here, if you mutate early on in the guide, so if you target the DNA, the PAM is here, so you'll find the PAM here. We'll call these bases PAM proximal. If you make a G to U substitution at position 2, for example, this becomes white, so it means there's no cleavage. So Cas9 nuclease cannot tolerate that mismatch. However, back here you can see a lot of blue, meaning in general these bases were really not very specific. They did not terribly contribute to specificity. If you look across multiple genes, you'll find that in general there's this runway of white before you get to blue. And that seemed to suggest this seed sequence for specificity, rather analogous to RNAi, where only the first 10 bases of this guide sequence seem to really contribute to specificity. So that is a general model that fits the single base mismatches. If you do multiple mismatches, the story gets altogether too complicated. I won't show that data here because it's difficult to kind of really understand it, but what I will say to summarize is that all bases within the guide RNA contribute to different degrees to Cas9 targeting specificity, and this is dependent on the quantity, the position, and the identity of those mismatches. Okay, and this would allow us then to try to predict, given a particular guide sequence, what are the off-target sites throughout the human genome? So you can do this by computational prediction. So if you have a particular guide sequence of interest, let's say this targets the gene that you want to target, then you can predict sites throughout the human genome that have mismatches compared to this guide RNA. So what we were able to, based on this in silico prediction, find off-targets that had high rates of off-target modification. Okay. And so this was really interesting because then you can start to imagine there are ways of selecting guides that may be better or worse than others. Okay. And eventually we collapsed all this into an open source online tool that I'll describe shortly. However, we were first really asking ourselves, given this kind of specificity, right, if you have an on-target modification of almost 50%, an off-target modification of almost 20% is really quite poor. So how could we make this better? And so it turns out that specificity is tuned in part by the enzymatic concentration. The amount that you deliver is really important. And this really makes a lot of intuitive sense, right? If you really flood a cell with only two really potential true on-target sites, then you could start to decrease the specificity of recognition. And this indeed seems to be the case. So here on the x-axis, you graph the dosage of these plasmids that you transfect. And essentially you see as you increase the dose, your on-target modification goes up. However, if you graph the on-target over the off-target rate as a specificity, you see as you decrease your dosage, your on-target over off-target rate starts to shoot up. So by titrating the dosage of these Cas9 reagents you introduce, you can start to improve specificity to a certain extent. However, this is okay. This improves by maybe 5 to maybe 35-fold, maybe 7-fold improvement in specificity, but that really just wasn't good enough, right? So how can we really engineer this system to make this a more specific mode of recognition? And so our inspiration came from the previous generation of gene editing tools, zinc finger nucleases and tau effectors, where they have two different tails. These are DNA binding domains, both of which bind cooperatively at your locus of interest. And they recruit two different halves of a flock1 endonuclease that will heterodimerize to generate a double-strand break. So then recall that previously I said DNA nicks are repaired with high fidelity within the cell. But what if we could do multiplex nicking, rather double nicking, where we nick the top strand and the bottom strand of DNA simultaneously? This would theoretically mimic a double-strand break, whereas any off-target sites by this nickase Cas9 and this guide RNA would be nicked. And so those would be repaired with high fidelity, and so then you would have only DNA double-strand breaks at your site of interest. And so we tried to investigate whether these theoretical double-strand breaks could actually lead to indels, and it turns out they do. And in fact, you could get really high indel formation, up to 40 to 50%, so quite comparable to the wild-type nuclease, okay? And then we asked ourselves, how far apart could these guide RNAs be technically to be able to generate a double-strand break? And we found if you define the distance between these guide RNAs as the guide RNA offset, a negative offset then would be dragging these complexes together. So you can see from the data that typically that just doesn't work. And you could imagine, because then if you drag them together, these two complexes would not be able to bind at the same time. They should really sterically clash, and that's why you get no indels. However, what was quite surprising was that by dragging the complexes really far apart, even with almost 100 bases or so of intervening sequence, you could still get indel repair. And while this does not make much thermodynamic sense, functionally this led to indel formation. And so that's really an interesting and outstanding question. But by sequencing, now we ask ourselves, we can generate double-strand breaks by Cas9 nickase multiplex nicking. Does this really improve the off-targeting specificity? And so by doing deep sequencing with Illumina, we are long story short, be able to reduce the off-target activity by over 1,000 to 1,500 fold, of course, depending on which target site you're interested in. And because these double nicking experiments allow you to generate double-strand breaks, we were able to show that it works to generate modified mouse zygotes for making more specific kinds of transgenic loss-of-function animals. So we took all this data together and generated a computational tool to identify guide sequences. So this is probably a slide that you'll all be very interested in if you want to use CRISPR. So you can find this tool at tools.genome-engineering.org or simply crispr.mit.edu. And basically what this tool does is it allows you to input your exon of interest. So let's say you want to modify in your cell line or in your animal a particular genomic locus. You can take that exon of interest and input it into the program, and then it will identify automatically what all the possible guide RNAs could be. And then it uses a data-driven computational algorithm to score each individual guide RNA depending on its predicted specificity throughout the genome of interest. In this particular case, it's a human genome, but now we have support for almost 20 genomes available. And so essentially, see here, we score the guides so that you can basically figure out which ones would be the most specific. So that's efficiency, and now we've talked about specificity. Let's talk briefly about inducibility, novel effectors, and orthogonal control or multiplexing. So it turns out if you can catalytically inactivate one catalytic domain of Cas9 to convert it into a nickase, what if you did it for both of those to make it a catalytically dead Cas9 or dCas9? That becomes a generalized DNA binding domain. Then you can then just simply fuse these to different functional effectors to recruit those functions to your locus of interest. And this allowed us to start to make designer transcription factors, where if you have a Cas9, catalytically inactive Cas9, by fusing it to different effector domains, VP64 is a synthetic transcriptional activator, SID4X is an engineered repressive domain. You can then activate or repress DNA, your locus of interest at will. Okay. There's another epigenetic modulation proof of concept. So we did a similar paper where we took tau effectors, which are DNA binding domains, and fused them to different kinds of epigenetic enzymes. So here you have a bunch of histone effector domains. Here we illustrate histone deacetylases. So a histone acetylation is typically thought of as an activating epigenetic mark. So a deacetylase then should repress the gene transcription. So here we show fold reduction in the mRNA. So by targeting your particular locus of interest with tail H-stacks, or these are all different fragments of repressive domains from epigenetic complexes, you're able to really repress genes and modify the epigenetic regulation at your locus of interest. So we're really blowing by here. Next, Cas9 homologs are functional mammalian cells. So you can imagine you have one Cas9 in a cell that targets a particular guide. If you put in a different Cas9 that has a different PAM, it could then be completely orthogonal in the sense that it could target an entirely different DNA sequence of interest. So by introducing libraries of different Cas9s, you can really expand the toolbox of possible functions by doing orthogonal control. So you can imagine in one cell, you can turn on one gene and turn off the other gene. Or if you have a heterochromatin state, you could try to modify that and make it into euchromatin, make it into a more actively transcribed region. And so this is really exciting because it has really broad applications for a variety of experiments. One other thing we did that Ophir will talk about later is genome scale CRISPR knockout screening, where essentially you can, in a big population of cells, knock out every single gene throughout the human genome. This would allow you to really do genome scale screens for cancer, for neural development, for anything really. So one really interesting thing that people have been doing for a long time has been RNAi and ORF screens, right? And one exciting paper based on these CAR T therapies or chimeric T cell receptors has been to try to generate different T cell receptors that can specifically target a particular cancer in a patient. And by doing these genome scale screens in these T cells, they're able to figure out particular pathways that can be manipulated to generate better cancer therapies. And so this is one area that is really exciting that we'll hear about more. Now I'll touch briefly on structure-guided engineering. We have a Cas9 crystal structure. What does this really mean? What can we do with it? And so let's take a little closer at the Cas9 nuclease. It's really quite confusing, but really I'll orient you. Here in purple we have the H and H domain, and here in blue we have the RUB-C domain. So these are the two catalytic domains of Cas9. And here in yellow is the target DNA that is base paired to the guide RNA, which is in blue and then red, wrapping down here. And essentially, then you can find that there are these two major lobes. This is a bilobe protein when it's bound to DNA. It has a top lobe and a bottom lobe that together sandwich the RNA and DNA heteroduplex together. So if we take a look at a surface space-filling model, this gives us a more intuitive sense of what this looks like. It essentially just clamps around this heteroduplex. And one question we had was, how does the Cas9 recognize this PAM? How do different Cas9s throughout the microbial diversity be able to target different PAM sequences? And we identified this C-terminal region as being responsible based on the series of chimeric mutagenesis studies. And so there is a Cas9 ortholog from S. thermophilus called ST3-Cas9 that targets the PAM NGG, NG, okay, while the S. pyogenes Cas9 targets the PAM NGG only. And so if you take the ST3-Cas9, this is ST3-Cas9, you find that it can target sequences with its PAM. This is NGG. However, it was not, NG is not detected, it was not able to cut DNA with only an NGG sequence. Of course, the wild type Cas9, which targets NGG, can target NGG, and also the NGG within this longer PAM site. So by taking the C-terminal region of SP-Cas9 and chimerically adding it, replacing it with a C-terminus of ST3-Cas9, you're now able to target NGG PAMs alone. And so this is really exciting for a variety of applications because the S. pyogenes Cas9 is actually a very long sequence. It's a very large protein. So typically when people do in vivo applications, they like to do viral packaging to deliver an effector protein. So however, the people typically use AAV, adeno-associated virus, which has a 4.7 kb packaging limit. So this is a very technical problem where it's very difficult to package the Cas9 nuclease into adeno-associated virus. AAV, however, is really desirable for in vivo applications due to its low immunogenicity and its really well characterized array of serotypes for infecting different types of tissue. It's also being used in the clinic in various clinical trials for various kinds of applications. So it's a really attractive way of doing in vivo delivery. However, Cas9 is really quite too large. So one experiment you could imagine doing, okay, is mining shorter Cas9 orthologs that are shorter, but these typically turn out to have longer PAMs. So then by doing these sorts of experiments where you take a shorter Cas9 and you replace the longer PAM with this more relaxed NGG PAM, you can start to engineer different Cas9s that have optimal function for in vivo applications. We're running short on time, so I'll skip a couple slides. The last one I want to talk about is the REC2 domain of Cas9. So if we go back to the space-filling model, you'll see that these REC domains, which stand for recognition, of course, are involved in recognizing the RNA-DNA heteroduplex. However, you see that this REC2 domain kind of pops up out of the crystal structure. It actually doesn't have direct contact with the RNA-DNA hybrid. And so by truncating this domain, which by the way is variably conserved in shorter Cas9 orthologs, you're able to generate a truncated version that is much shorter but actually still has significant on-target activity. Interestingly, although the indel rate here is lower than the wild type, by Western blot, the protein expression is concomitantly lower. So this could be due to some kind of misfolding. And this can be continuously optimized. So instead of just taking shorter Cas9s, you can figure out ways to also truncate the current ones that we have. So these are some of the really important things that we're working on in CRISPR technology development. And of course, these will really inform the kinds of applications that we're able to do in the future. With that, I'd really like to thank you all for your patience and your attention, and to acknowledge my PI, Feng Zhang, the various members of our lab, and our collaborators, as well as our funding sources. So I'm happy to take questions.